Chapter 6 provides an overview of the structures and function within mammalian, plant, and prokaryotic cells. Three objectives in this chapter are: 1. To be familiar with the regions of the cell and the overall role of each region, to earn a basic familiarity with basic organelles and their function, and to become familiar with the way neighboring cells can communicate with one another. All living organisms are made of cells, of course. There are two basic types of cells, one of which is known as a prokaryotic cell, and that which lacks a membrane-bound structure other than a plasma membrane. In other words, it has no membrane-bound organelles such as eukaryotic cells do. Eukaryotic cells, on the other hand, contain membrane-bound organelles which enable what is known as compartmentalization, and this leads to specialization in functions within those various organelle structures. Examples of eukaryotic cells would be those found in animals, plants, protists, and fungi. There are certain limits on cell size, and this is determined primarily by its ability to perform metabolic processes. So on the lower end, we look at just all those things that are necessary components to allow some of the most basic functions to fit, and in the upper regions, those which can promote more advanced functions and regulate supplies in an adequate fashion. There are three main regions of a eukaryotic cell, and that would be the nucleus, the cytoplasm, and the plasma membrane. Here we look at a comparison of the two general type of eukaryotic cells. At the top you see an animal cell here with the nucleus, composed of a nuclear envelope, the nucleolus, and the internal residing chromatin, along with some of the other internal structures, compared to a generalized plant cell with some of the same basic features with the exception of a large central water vacuole which controls water pressure in plants. The nucleus is the first region, serves as a control center of the cell, and of course includes a chromatin at its core. The nucleolus is a region within the nucleus in which RNA and some more specified hereditary material reside. The nuclear pore structure is such that it allows materials to enter and to exit the nuclear region of the cell. In order to allow materials to enter the nucleus, there must first be a 27 amino acid tag, known as a zip code, that is added onto the molecules that wish to enter the nucleus. And these molecules are assisted by proteins known as importans. In order to leave the nucleus, as you can see here on the bottom diagram, exportin proteins, a specific zip code or address by which the materials are transported to, and a GFP and RAN protein are required for this to occur. Here's just a summary of the way proteins are imported into the nucleus. First of which occurs is that an NLS, or nuclear localization signal, is attached to the incoming protein. When they bind together, they will then enter through a nuclear pore structure, which is in the lighter green here, and coupled to this is a RAN or a RAN-GTP molecule, which provides the energy necessary in order to facilitate this process. Once the protein has been imported into the nucleus, the protein dissociates from the RAN-GTP and the NLS signal, both of which then exit the nuclear pore, and through a process of hydrolysis, the GTP is hydrolyzed into a GDP, and the important protein here, seen in the green, dissociates from that and can function to return its function and allow more proteins to enter into the nucleus. Ribosomes, of course, are simply sites of protein synthesis and reside in either bound or free formats. The endoplasmic reticulum, part of the endomembrane system, functions in two ways. The smooth ER serves as a site of lipid synthesis, detoxification, and carbohydrate metabolism. Rough ER, on the other hand, simply the site of protein synthesis, and that's all it does all day long. The Golgi apparatus functions in the sorting and the packaging of manufactured products of the cell and transports it to its various regions. There are two regions on the Golgi apparatus, that of the cis, which receives products from the endoplasmic reticulum, and the trans portion, which ships materials to other parts of the cell. Lysosomes are another structural support and contain hydrolytic enzymes serving as the cell's stomach in order to degrade food, materials, or other foreign debris. Vacuoles are the membrane-bound storage area for various resources. They can either store water or other types of materials. Mitochondria and the chloroplasts tend to supply the majority of the energy that the cell requires to function. Peroxisomes house hydrogen peroxide and facilitate in the process of getting rid of foreign invaders or other types of materials that enter the cell that may harm the cell's functioning. Here you can see a couple of different microscopic views of animal cells. This is a scanning electron microscopy image of animal cells going through the process of mitosis. Here you find two images, both transmission electron microscope graphs, that demonstrate the same process of mitosis, and here just a cross-section demonstrating all the various organelles. More importantly, when we look at some of the cytoskeletal elements, we find three major types: the microtubules, microfilaments, and the intermediate filaments. Both the microfilaments and intermediate filaments serve in the pulling forces which function during the process of mitosis, whereas the microtubules, as we will see in the chapter on mitosis, actually extend from the centrosome region, or a pair of centrioles aligned at 90-degree angles from each other, and send out these microtubule spindles that attach to the chromatin to allow them to enter the intermediary phase of mitosis, known as the metaphase stage. Cells also have organelles of motility, and they come in two varieties, flagellum and cilium, depending on their function. Some extracellular surfaces to be concerned of: the fact that most cells in the external core have a combination of proteins, lipids, and sugars, forming what's known as this extracellular matrix. Plants as well have an additional structure known as a cell wall that prohibits them from going through a process of lysis when too much water has been intaken by the cell. One of the ways that cells' functions are distinguished from one another, the quantity and the type of organelles, and in which case determines how the cell can function and to what degree. Communication between cells is also crucial, and there are various junctions that indicate these communication lines. First type is a tight junction. These are the leak-proof connections between cells. There are also desmosomes, which fasten cells together, strengthening the tissue. Gap junctions, which provide cytoplasmic channels between cells and allow cells to communicate with one another. And in plants specifically are structures known as plasmodesmata, which allow these same channels to be able to allow two plant cells to communicate with one another. In looking at tight junctions here, again we see a transmission electron micrograph of a tight junction. And we can see here various proteins that hold together the two surfaces of cells. And again, these serve as leak-proof connections between cells. Desmosomes. We look at an electron micrograph here of a desmosome. Again, here we see the sort of anchoring function that these proteins have, and how these membrane proteins actually allow a link to form between two neighboring cells, facilitated by internal filaments, intermediate filaments between the two cells as well, which extend inward into each adjacent cell. And finally, plasmodesmata. These are which create gaps that connect plant cell walls, in addition to the gap junctions, which function in the same way as plasmodesmata do, with the exception that these are found only in animal cells, but they allow for channels to be formed between two neighboring cells, and again allows materials to pass and communication to occur between two neighboring cells. So that is all for Chapter 6. If you have any questions, please email me. Thank you.